Well, hello, friends. I think. I don't know if this is working. Oh, it's working, sort of. Okay. Well, well, that was frustrating. I tried to follow the rules and it wouldn't work. And so finally I resorted to the old Google Hangouts method, and I've got a message here that Hangouts on Air is going away August 1st. Oh, great. Okay. So I got to figure all this stuff out again. This does not make me happy. Oh, well. So it's working for now. So, hello. I'm sort of back. Some of you may have noticed, no, most of you probably didn't notice and didn't care. But I haven't been doing much on YouTube lately because I've been really busy. This has been a rough time of the year because all winter long I'm frozen in. There's a fair bit of free time because I'm on sabbatical, which is nice. And then the summertime hit, and I've got all these projects, and I've been booked solid with these things. So yeah, I've been playing with spiders. See, that's the real thing. Is this is the time of year when at least in Minnesota, when all the spiders are reemerging, and I'm trying to master a little taxonomy, and this is a new skill for me. So I spent the winter reading books, but reading books is no substitute because yeah, it takes hands on. You got to actually get in there and play with things and see things, and then you can start figuring it all out as you go along. So that's a lot of what I've been doing lately is going out, prowling around the yard, the house, the garden, checking out the town, looking for spiders. And yeah, there's spiders everywhere. And then trying to figure out who they are, which is a non-trivial task to do. So yeah, I get home in the evening and then I go play with spiders again, just trying to sort out what I've collected. And there's some really cool stuff out there I'm discovering. It's like a zoo or like an alien planet outside my house. So we found some tetranatha the other day. We've got lots of jumping spiders. We've got these creepy, weird volcids. We've got the theridaidids everywhere. That's the one I mainly want to study. So it's been just tracking those things down and figuring out what they are. Then on top of that, last week I went off to this meeting. Is that reversed for you? It's reversed. Well, anyway, this is the meeting of the American Arachnological Society. It's the 48th annual meeting, and it was held in Lexington, Virginia, at Washington and Lee University. And so I went to that, which was a novel experience because, I discovered very much what imposter syndrome is like when I was at this meeting because I've been working on zebrafish for about 40 years, believe it or not. And this is the year I've decided to go off and learn something completely new. So one of the reasons I went to this meeting is to get exposure to people who do this stuff and see what kinds of things are conventional in the field and what people are expected to know, and discovering that I don't know 99% of what I need to know. So I've got work ahead of me. So that's what I've been working on. And it was a strange experience because I'm 62 years old and I felt like a first year graduate student at this meeting. So that's kind of refreshing, right? that's an invigorating experience to be this old and then to discover yourself all over again and learn new things and be overwhelmed by all the data. So yeah, I spent it was a 4-day meeting. I was there the whole time. I attended every session like a noob. Sat through everything and struggled, and it was a very diverse meeting because they talked about lots of other things like, oh, well, I learned all about opiliones. I learned about solifuges. Uh, there was a fair bit about megalomorphs, which we don't have around here. So it was a couple of days of exotic biology as far as I was concerned. I mentioned earlier, it's kind of like going to an alien planet, and that's kind of exciting, actually. All these weird things that are out there. I really recommend it. The other thing is, I've been finding that you remember when you were a kid and you were really into dinosaurs and you started reading all about dinosaurs and you memorized all these exotic names, and it was trying to figure out all these exotic relationships. Uh, that's kind of the situation I'm in now, only it's spiders. So yes, some of you may have noticed on the blog that I've been a little bit obsessive about spiders, and that's because that's my reality right now. Is I'm out there looking at spiders. I got to catch them all. Yep. So it's fun stuff. Uh, let's see. What else do I have to say here? The projects I've got going. I've got a couple of projects going right now. So we've just finished setting up a greatly enlarged colony in my lab, thanks to my student, Preston Pfeifferak, who's been helping me out a great deal. So we've now got, a dozen cages of spiders. One of the things we learned from some of the experts is they need more room. That I was keeping them in these little vials for a long time, which is fine. They thrive in there as long as they're alone, but they're really cramped in there. And when you throw in another spider, they don't know how to react. Imagine that you've been thrown into solitary confinement. You're in a little 8 by 10 room and you don't get out. You don't get to see anybody. Somebody throws food at you every once in a while. And then all of a sudden they throw in a gorgeous member of the opposite sex. You're not going to respond in a normal way. So that's probably the reason we've been getting some mortality in our males is you got a female living in this cage and you throw a male in there and they don't know whether it's prey. They don't know whether it's a hostile enemy. Uh, Having sex is low on their list of priorities, and so we end up with a lot of mortality. So what we've done is we've got these, they're like the size of a large shoebox, these plastic containers, and we put in cardboard frameworks inside so they have something to clamber around on, so the environment is a little bit more complicated. And we let the spiders acclimate in there, put a spider in, let it sit there and be happy for a week. And what the females then do is they build these nice cobwebs all over the place. They're very pretty, actually. I should try and get a picture of some sometime. Really complex three-dimensional webs that sort of fill this space. And then when you throw in a male, they don't freak out, and the male has some place to run away to. So it's actually been fairly productive that way. This past week, like I said, we got about 12 of these, and we threw in a bunch of males on Monday. And they're all alive now except for one. So that's good. And the one was a weird situation where we threw the male in, and instantly the female just went berserk and chased after him and gave him no rest. He was able to run away and hide, but she eventually caught up to him a few days later, and yeah, she ate him. It was tragic, but that was an unusual situation. I don't know what was going on there. Spiders have personalities, and this one was just kind of evil. Um, by the way, we're naming the spiders. Preston is naming them after Game of Thrones characters. So Sansa is the one who is really bitter and angry and vicious. Oh, don't know what that means. Anyway, so they're all sitting in the lab right now in their nice big roomy boxes with a consort. And we're hoping we will start seeing some little baby spiders in the near future. We also have, this is pathetic. We've got one egg sac right now, just one. What we need is like, 3 or 4 or even half a dozen a week, and then we can start doing experiments. So what we've got is this one, and it's not even the species I want. It's uh, Steatota triangulosa. We found one of these. It's a related species. We found one of these in a garage around here, and we just took it back to photograph it. And then she goes and lays an egg sac on us. It's been sitting there for about 2 weeks, and today we looked, and yeah, there's starting to be some little spiderlings in there. So that's a 2-week developmental time. Parasteatota is supposed to be 8 days. We need to get some so we can test it. Anyway, so the projects we have in mind right now is we got this breeding colony going. We want to get regular collections of egg sacs and lots of embryos to play with, because then we can start doing some developmental biology. The first project we have in mind is to do a developmental time series. There is a published time series already, so this would not be new. The one thing we want to try is changing temperatures and see how the embryos respond to variations in temperature, whether there's a lower limit where they go into diapause, or whether there's a lethal lower temperature, how the rate of development varies with different temperatures. This is what we're going to tinker with over the course of this summer. So we'll see how that goes. The other thing we want to do is, I have a son who lives in San Antonio, and I haven't told him this yet, but I would like him to collect some San Antonio spiders for me, because there's a paper out there that reports that southern spiders are more sensitive to temperature changes than are hardy northern spiders, even of the same species, which I think is kind of cool to have this kind of cline of developmental variation within a single species. And so I just want to get some Texan spiders and compare them to Minnesota spiders. And sorry, Texans, I predict that Minnesotan spiders will be much, much more resilient. We'll see. It may also be possible that what we'll discover is that Texas spiders are better able to handle higher temperatures. I don't know. Lots of things to tinker with there. So that's our first lab project. Is let's grow up some babies at different temperatures and quantify their response to variation. Then the other big project is ongoing all summer. Once a month for a week, we go out and survey various houses in the Stevens County area. Really, it's mainly in Morris, and we've been checking out these places. And that's kind of a blast because, we get permission, of course, and then we go into people's garages and we scout around. And right now, we all have extremely well-tuned eyes to detect spiders. So we go into these places and look around, and you would be amazed how many spiders you can find in a garage. Sometimes we find a garage that somebody's been very diligent in cleaning out, and then maybe we don't find very many. We still often find some, but not very many. And then we found some houses that they look like they're about 60, 70 years old, and the garage has never been cleaned out. There's just sheets of cobwebs everywhere. That's our favorite kind of garage. And then you find lots and lots of spiders there. This is the one problem, the one limitation we're having so far is we've artificially constricted ourselves, and we said we're only going to look in these garages and sheds, which is kind of a uniform environment. And what we've been finding is that the species of spiders that are present are also pretty predictable and uniform. That what we're finding is just we either find Fulcus phalangioides is the most common one there, or we find Peristeatoda. And occasionally we find things like Steatoda triangulosa. Uh, we've found a few Steatoda borealis, things like that, related species. But those are the ones that seem to be best adapted to these kinds of synanthropic environments. And we've just been counting them. Sometimes we find lots, sometimes we find a few. That was in early June. We're kind of curious to see what things will be like in early July, because temperature's been going up, and even just my casual inspection around the house, I'm finding lots more spiders here. So that may be data. We'll see. We'll see how the numbers change in the next. We'll be going out in 2 weeks. We'll check it then. And we're going out in August. We'll check it then. And then I'd like to do another survey in early fall. The catch is that I need student labor to help me out. And the students are going to be taking classes. Suddenly they're going to have a different priority. So anyway, yeah, they can't just be my lackeys all the time. No, they're not really my lackeys, but they've got lives. I have to respect that. Darn it. So that's where the project is right now. That's what I'm doing, is thinking spiders all the time. I know it shows, but it's fun stuff. Let's see, what else have I got on the books here? Oh yeah, next week I'm going to be at Convergence. So planning to attend this big science fiction convention in Minneapolis. That's next Thursday through Sunday, and I'll be there the whole time playing around. I have a costume, sort of. Yeah, they on Saturday they usually have the big costume event, and I'm just going as me, but I'm going to wear all my spider gear. Yeah, I've got, it's not handy here, but I've got my goggles. I've got a bandolier that's full of these little tubes that I use to collect spiders. I have a hand lens. Yeah, I'll look very nerdy, but then I always look very nerdy. That's my nature. Let's see, then what else? After that, who here has been following Iris on Death to Squirrels? She's got one of the blogs on Free Thought Blogs. She's a lovely lady. We got together in New York, was it last year before, when we went off to participate in the revolutionary communist protests about the Trump administration. She was very eloquent and was working hard there. She also writes the blog Death to Squirrels, which has been kind of moribund lately, and there's a reason for it. She's been getting cancer treatments, but apparently she's on the mend. So that's the good news, okay? So we're all happy to. I'm happy to report, and I'm sure you're all happy to hear that Iris is doing much better, and she is flying out to Duluth for a vacation with her husband, and we're going to drive out and meet her there in Duluth and maybe have a nice dinner, maybe check out some parks and scenic areas, all that kind of stuff. So that's on my agenda. Let's see, then what else after that? Well, it's just lab stuff. Then Skepticon in August. Who's going to be at Skepticon? Everybody should be at Skepticon. It's a really good conference. And I know some of you can't afford it. It's a free conference, but even so, you got to get to St. Louis, uh, and you got to stay there for a few days. There's lodging costs and travel costs. So no, I don't mind if you can't make it, but it sounds like it'll be a good conference. Skepticon is one of those few atheist conferences that all along has really been into diversity and equality and social justice and all that great stuff. So if you want, you can go there. Oh, let's see. There's comments here. Ah, yeah. Let me just answer some of those things. Daddy longlegs or harvestmen. That's right. They are not spiders. They are opiliones. They're easily recognizable and distinguished from true spiders because they don't make silk, for one thing. They're not venomous. And their body, spiders are two segments, cephalothorax and abdomen. And opiliones are just kind of fused it all into this little ball of a body. So that makes them pretty obvious. Um, yeah, they're cute, but I work with real spiders. None of these fake spiders. No, it's all good. Uh, let's see what else is people. What else is it true that free speech warrior Richard Dawkins won't show up unless they deplatform those crazy feminists? Uh, I have limited knowledge of that. I don't know whether it's universal, but I know that there have been a few cases where he has blacklisted women from speaking at the same event he does. Talk to Rebecca Watson if you want to get the whole story. Although I get the impression that Rebecca Watson is really, really tired of all this stuff and is happy not to be associated with the atheist movement anymore, and is glad to move on. Speaking of which, I was at this, as I mentioned, I was at this conference, which was great. Also at this conference, the weird thing is most of the people don't know who I am. That's not weird. That's kind of to be expected. So I was kind of a cipher there, except that there were a few people who did know me from zebrafish stuff and from my atheist activism. And I had a few conversations with those, and it was very interesting. Several women who said they had been active in the atheist movement years ago, and they left. They stopped following me too, and they stopped following all these atheists. And why did they do that? It's because there were such assholes running the show. And, you know, this kind of breaks my heart because I invested so much time and effort in the atheist movement, but it was all to no avail because I left them, they left me. We kind of split, not very amicably. And what I keep finding is these people who would have been so happy to be working with atheists and for atheist causes, and who had been disenchanted because atheists didn't want them because they were women or minorities, and there was no prospect that they'd ever be put into a productive role within atheism. So they gave up. Yeah, I kind of gave up too, I'm sorry to say. Anyway, let's see, what else is there? Any other questions? People have any other comments? I've been babbling for half an hour. Let's see, what else is going on? I forgot to mention that after all these things this summer, what's going to happen is I have to start teaching again. I'm going to have to go and remember the stuff I teach, I know I remember fairly well, but I got to get my act together. I got to get my office cleaned up. I got to prepare my syllabus. Uh, there were a few changes that were made when in the classes I teach by my colleagues, so I've got to adapt to those. And if any of you know about real teaching at the college level, you got to keep up with all the material. So I, I feel like I've been keeping up with that stuff, uh, but I just got to make sure I've got it all updated for my lectures. So that's coming up real soon. Yeah, my sabbatical is coming to a screeching halt, and I'm going to panic. Nah, I'll cope. I've been doing this so long. I think I can handle it. Let's see what else is happening. Oh, I've also got to get my act together and resume Cafe Scientifique. I've been the organizer for Cafe Scientifique for many years here in Morris, um, and I've got some ideas for some new changes I'm going to implement in that. One of the struggles with it, the whole purpose is get faculty out into the community and doing that community engagement thing and talking to people from the town and listening to people from the town and getting questions from them, which is all great stuff in principle. But often what I've been finding is we get out to that we get the meeting together, we get people there, and then our speaker is doing the old, let's lecture from PowerPoint again. And um, that's a good, dense way to get information across to students who are obligated to be there. And it's not such a great way to communicate to the public at large, that there should be some more give and take. So, um, yeah, so we're going to do some format changes to force people to actually talk with rather than at people. So that's coming up. See, my plate is kind of full right now. Let's see, another question. Do I think I'll get a beer with Richard Carrier at Skepticon? Why, no, because Richard Carrier has been banned from Skepticon. He will not be there. I can't imagine he's going to show up. So, no. And I don't think we're even going to say much about him. We might do a little strategic discussion with the defendants in his lawsuit, but otherwise, no, I'm planning to have a good time there. And that's not fun. Let's see. Why spiders? How about snakes? There's a rattlesnake in the north as well, right? Sure. Um, we've got a couple of people at my university who study herps. So Heather Way and Rob Denton are two faculty members who study various herps in this area. And yeah, there's diversity and there's cool stuff to be learned from them. Rattlesnakes, I don't think we have rattlesnakes here. I think Heather's been looking at garter snakes, and Rob has been looking at various amphibians like salamanders. So no, that's clearly too crowded a field. There are two people working on herps in this area, so I can't possibly do that. Why spiders? Because well, for some reason, I've always been attracted to organisms with a multiplicity of limbs. I don't know why I was wasting time with zebrafish that have no limbs. But yeah, cephalopods and spiders have something in common. All those legs all over the place. It makes them really cute and adorable. But spiders I'm mainly interested in because I drifted into this particular subject because I've been gradually evolving a greater interest in eco-devo, ecology and development, and how the environment influences development. And there are some things about zebrafish that are great for that. Having a model system where you've limited the variation and basically what genes you've got to play with, and you've also got total control of the environment. That's kind of a cool system to work with when you're studying this kind of thing. but there isn't much accessible natural variation. I'm sure if I went to Nepal or India and the native ranges of the zebrafish, I would find all kinds of cool stuff to study there, all kinds of variation in zebrafish and related zebrafish species. But with spiders, the variation is all right here in my backyard. That's what's blowing me away is once you learn how to recognize these things and how to spot them, uh, seriously, you get down on your hands and knees in your backyard and you creep along the boundary of the fence, or you look at the wall, the base of the walls. You go into the garden where you've got different things growing, and you discover this amazing world of all kinds of differences and all these competing species, interacting species, feeding on different kinds of prey, all of different sizes. Yeah, that you've got something like, these little these eastern parson spiders we've got there. They're really tiny little guys, and they just scurry around the ground looking for little things to eat. And then we've got these furrowed barn or no barn funnel spiders, which are maybe this big. They're huge. If you are just, if you're a civilian and you run across these, you freak out and are terrified because they're huge, but they're pretty much harmless. Um, so we've got those. Those build these nifty little funnel webs, and we found a few of them in garages around here where you look around and you see the usual cobwebs everywhere, and then you find this one little corner, and it's like there's this little tunnel of webbing, and you can follow that back in. And when you look at that, what you find is these uh, funnel web spiders that are just huge and amazing and really kind of cool. And before, I would never have noticed these. So what it means is if you're interested in interactions with ecology, and if you're interested in how the environment influences these organisms, spiders are a pretty good choice. I can think of other animals that would also be good choices, like wasps and ants. Ants kind of restrict their environments, so they're a little bit more uniform. But there's a lot of variety in ants. Um, Lots of things like that that would be good to look at. Birds as well. I mean, I could see I could see studying birds for this sort of thing, but um, there's more variation within spiders. And another plus is nobody complains if you do experiments on spiders. So that's something. So 8DX says, I'm kind of waiting for PZ to work on the differences between spiders of the same species at home. Yeah, that's kind of the interest I have is um, I'm doing this survey of all the varieties of spiders in this area right now, but I'm going to be focusing on Peristeatoda tepidoriorum, which is kind of the model system for studying spiders now. And I'll be working with those in the lab. And specifically, what I want to do is look at some of that variation. I mentioned things like temperature sensitivity and how they vary in that. The other cool thing about them that we're noticing is you stare at them and you discover you can start telling them apart. Yeah, you may say, all spiders look alike to me, but no, even within the same species, what I'm seeing is we're seeing little variations in pigmentation. We don't know how much of that is genetic and how much of that is just environmental noise. Uh, we've got plans in the long term to dissect some of that stuff. I'm also seeing variations in behavior, and there is apparently an active field of study on spider behavior, and specifically looking at what you might call spider personalities, that they have different responses to threats. There was a whole session on it at this meeting I went to. Um, So that's another cool thing. That's something I think a lot of people do not appreciate is that animals aren't just, oh, they're all the same. Even with zebrafish, when I was working with zebrafish, one of the things I was interested in is the fact that, gosh, you can detect individual differences in behavior in even zebrafish. Some are more aggressive and territorial, and others are more passive. There's all kinds of things that could be worked out there. I am asked, what is the largest spider in your state? The wolf spider? I'm not sure. I don't know what the largest spider is. Uh, it might be Dolomedes, the fisher spider. I would have to look. Yeah. See, I'm still learning things. So where are we going to work on? Let's see, Master. You thought you were going to be cutting up or poking embryos, so you see behavioral observations as more interesting. That's part of the story. I am going to be uh, working with isolated embryos and studying that as well. So we're gearing up for that as well. We just need more embryos. Once we get routine production of lots and lots of egg cases, we can start doing these observations. So, for instance, what we do is you can easily remove spider eggs from their egg sac. You can put them on a microscope stage. We've got this stuff called halocarbon oil. You can immerse them in that. One of the things I have to work out is how long can you keep a spider embryo alive in halocarbon oil? Because it would be cool if we could keep it alive for a day or 3 or 4 and actually do some time-lapse recordings of the development and figure out what's going on there. So yeah, we're gonna be doing that. That's gonna be stuff we're mainly gonna start doing that in the winter months, I think. So this is summer. We got all this wonderful diversity of spiders out there. So we're kind of exploring that while we can. And then once it freezes in and the snow falls and all the spiders retreat and go into diapause or die or whatever, then we'll see what we can do in the lab. Uh, and that's also another of the virtues of this system is we can do field work in the summer and then we can do lab work in the winter and get a fairly rich research experience for my students, which is kind of the priority. I'm an old guy. I'm sort of out of it and done, and I'm not going to make any radical discoveries. But the nice thing is that we can still train students, and so the idea is we'll get students involved. Okay, other points brought up here. Would I want a brown widow egg sac next time I find one? Ooh, that would be neat. Yeah, sure. Send them to me. Um, Yeah, when the question would be, are you going to send those? Uh, let's see, one way you could send them to me is, uh, to protect the postman and all that stuff is if you just got like a pill bottle or something, an empty one of those, throw them in there. You don't have to worry too much about poking holes in lids or things like that because spiders have really low metabolic rate. And if it's taken a few days to get here, it'll be fine. It'll be just fine. Um, yeah, so that would be cool. If you were going to do it though, you'd have to, oh yeah, test tube would work too. Just put a cap on it. So, I don't want to be held responsible if a bunch of brown widows get loose in the post office. So seal them up good. But they have low metabolic rates and they'll be just fine. Uh, you do have to send them over the summer because winters here, it doesn't work. We have had some miserable experiences where we try to order live animals in January or something. Um, and all it takes is that they get left on a loading dock for a day, and what you've got is a cube of ice with frozen animals in it. So, yeah, we stopped transporting things once the freezing weather hits Minnesota because they're not going anywhere. All right. There's an invasive species of tick in the eastern U.S. that's more likely than the native species. Yeah, ticks. I don't like ticks. I don't like ticks and mites. Um, yeah, they're related to spiders. They got 8 legs, all that kind of stuff. Uh, but they're the nasty parasitic cousins, and I want nothing to do with them. I do have a colleague here at UMM, Tracy Anderson, who is specifically a freshwater ecologist, and she studies ticks and mites. And so I'll let her have those. I want nothing to do with them. Yeah, yeah. We had a shock when this earlier this summer. I was playing with a Burmese funnel and just taking a look as things were thawing out at what was in the leaf litter in my lawn. And yeah, we found ticks all over the place in my lawn, which surprised me. So I will keep that in mind in the fall, and I will not go jumping in a pile of leaves and roll around in it, because now I know what lives there, and it's not pretty. Okay. What else is happening? Have I talked enough? We're coming up on almost an hour. Trying to think what else is going on. Um, if we keep the news spider related, y'all know that Spider-Man Into the Spider-Verse is playing on Netflix. I could go watch that right now. What am I doing talking to you guys? It's my favorite. It's my favorite superhero movie. Is that one? Really well done, really creative, really pretty. So that's fun to see. Next week is what is this? The next Spider-Man movie that's coming out. I'm sure it won't be as good as Into the Spider-Verse, but it'll be fun. Yeah, yeah. So I'm planning to go see it. It's going to come out here. It's going to be at our local theater, so I can go see it. I've actually offered to my research students that I'll take them there if they want to go see it. So, yeah, Far From Home, that's what it's called. Uh, the previews, it looks like more of that nonsense of superheroes and supervillains destroying villages and cities, in this case, somewhere in Europe. Um, okay. Another question. Have you looked how climate change has or might change the types of spiders that live in Minnesota? We haven't yet, but that's on my list. So the whole purpose of the spider survey is to get a baseline data on what spiders are here now. It probably would have been smarter if I'd started that 10 or 15 years ago, but hey, I'm new. Okay, I'm new to this. Can't you tell? Look at this. Look at this childlike face. I'm brand new. Um, so yeah, we're getting baseline data, and that's one of the things I propose the university is, in the long run, we'll repeat this every year, and we'll get a feel for how spider populations respond to changes in climate as they happen. Maybe there's a lot of variation in weather, though, so that means we got to keep doing this for years and years. So apparently, I have to live to be 100 years old and still be spry enough to go look at spiders. So that's the downside. But okay. So yeah, we're going to do that. And I'm also saying that spiders are a good proxy to look at things like variation in insect populations, because, of course, spiders eat insects primarily. So if you've got a feel for the variation in numbers of spiders, you've probably got a feel for the variation in the numbers of their prey. So yeah, so you've hit the nail on the head. Yes, we are thinking along those lines, and we'll be looking at those sorts of things over the next few years. Again, as long as I promise to live to be older, much older. I'm working on it. We'll see. Okay. Any other questions? Are we having fun yet? I'm glad this is working finally. Okay. Well, a question. I wonder if there's a spider analog for the Schmidt sting pain index. Hmm. That's an interesting. Hey, should I put that on my list of things to develop? I'd have to get myself bitten by spiders and say, but here's the thing with spiders are fairly innocuous. There's not that many species that will bite you. And the ones that will bite you, you really have to prod them to get it. They have no interest in you as prey. You are just an obstacle that's in their way. So it's a struggle to get them to work to do that. Also, most spider venoms are adapted for killing and digesting insects, not mammals, not mammals or birds. Although there are a few, of course, a few mygalomorphs that consume small mammals and birds. Uh, so yeah, most of the spiders, even if they bit you, you wouldn't notice anything unless you had something like some kind of allergic reaction to it. Uh, the spiders I'm studying, Parasteatota, actually do produce a venom that humans are sensitive to. They produce a cytotoxic venom that can break down. It actually causes necrosis in the cells, the bite site, and that's the main problem with being bit by these spiders. Is since they are causing necrosis, that means you've got an injury that is prone to infection, and most of the harm they do to people is by infection, not by the venom. Let's see. In 10 years, all your grandchildren can collect, prod, and dissect spiders, and you should let them know that next visit. Yeah, it's a little premature. Okay. My grandson is what, year and a half, and the youngest, my granddaughter, is she must be going on 8 months now. They're both adorable and rambunctious and cute, but they're not quite at the stage where they can start doing spider work. But give them time. Yeah, I'll work on it. As I compared it to dinosaurs before, for kids, I really think spiders have a lot of promise as a subject for kid-like hobbies. Uh, Another of the things I want to do is do some outreach in the near future and just meet up with kids and show them how they can collect spiders and introduce them to spiders and help them recognize the different kinds of spiders that live around here. Maybe I'll do that next year. All right. Let's see. Another point here. It's too bad you've distanced yourself from the new atheist movement. Some of my favorite videos of yours are yours. Well, thank you. Yeah. Uh, the thing is, even as a new atheist, a lot of what my videos were science videos. That what I was trying to do is describe, um, the reasoning behind the biology of evolution, and I think that's a useful tool for introducing people to atheism, and that's not going to change. I actually have plans if I get enough sleep this week. That's always a worry. I have a plan to put together a quick video this weekend on why there are more than two genders, and it's really a biological issue, and it just bothers me when people try to pretend that no, there's only two, there's only male and female. It's divorcing themselves from all the diversity in individuals and failing to recognize the complexity of the development of sex and gender. And I'm thinking I should say a few words about that, since it's another issue that's heating up in the atheosphere nowadays. What with the atheist community of Austin kind of blowing it in the last month or so. Um, Yeah, so that's actually a problem with the atheist movement in general, is that what you've got is a bunch of fairly smart people who get into the atheist movement and they think they've answered everything and that they're smarter than everyone in the world, and they aren't. None of us are, but they think they are, and they get cocky and arrogant, and they assume that because they are an atheist, they know the truth about everything. In some ways, they're kind of worse than evangelical Christians, because they perpetuate these distortions of the science. So yeah, what was that guy? Uh, the rationalism guy who kind of splintered atheist community of Austin. Uh, that's what was aggravating about him. He got up there and he pontificated about the biology of gender without knowing anything about it. And what he was doing is expressing his biases and using poorly understood science to justify them. Let's see. ADX says getting in some trans people to talk about the language that interacts with the science would be good. Yeah, yeah. As I mentioned, what I'm hoping to put together this weekend is just a casual video where I give an outline of my understanding of the biological principles that lead me to conclude that no, the whole idea that there's only two sexes is absurd. So I'll get that out there. But yeah, I agree. It would be pretty cool to organize or be part of somebody else's organized discussion of this whole issue. Uh, one of the things I have learned in my long experience with the atheist movement is that a little epistemological humility is a good thing, because I've seen so many people who lack it. I would say that in any discussion of trans issues, I am not the premier authority, obviously. I'm about as cishet as they come. Um, so that means that I feel like I shouldn't be the one to initiate a conversation about this issue. The one place where I do have some authority is in an understanding of the biology. And so I can see my knowledge being a complement to the real world experience of trans people, not to supplant it, just to be, okay, here's my biological understanding, how it fits with your real world experience, not that it overrides your real world experience. Okay, I get that. Oh, here comes the controversial questions. You don't think a person born in a male body has a physical advantage in sports after they transition to female? No, I don't. And you know, competitive sports is tough. People are always talking about and getting an edge. It takes intense focus and training to perform at the top of your field. And yet, here are these people who voluntarily, in a sense, in some ways, sociologically, it's not voluntarily, they feel like this is something they need to do, who have taken these hormones that changes their physiology in a way that makes them less optimal for male-dominated sports. So, no, I don't think they have a real advantage. And it's what annoys me is so much is people arguing that it's that there are men who are plotting to take hormones and get these surgeries even, also they can win a trophy. Yeah, right. You don't go through this experience unless you're really committed to that gender identity. Uh, so let's not penalize people for that. Okay, let's see. Yeah, he did a mea culpa, but yeah, the science isn't popularly known or communicated. No, the science is not properly known, and by a lot of people. And instead, what we've got is a lot of bias. As I mentioned earlier, people think they have all the answers because they know God isn't real. So obviously, they passed the test and are now qualified to lecture everyone on anything, including male and female physiology. Um, yeah, so as Joshua Strode says here, yeah, it's super complicated. Once you cut through the BS, it makes sense. They just don't have a real advantage. Yeah, these are people who are not doing this to get a competitive advantage in something as superficial as a sport. Uh, I know people care about sports, and that can be important to you as well. But they're actually changing their gender identity. You don't do that just because you want to score a touchdown or you want to run a little bit faster than the other people in the field. So now it bugs me that people are actually arguing about this. And there's a heck of a lot of people who seem to take this nonsense that that men are undergoing hormone therapy so that they can compete with women and beat them. The other thing here is that if you look at our culture, there is this strong idea that, oh yeah, it's no big deal if you beat a girl, right, in a competition. I, it, there's a lot of women who could run rings around me, but okay, there is this attitude that it's not real if you're competing with a girl, and so they're going through this for a non-existent virtue, for something that just earns them even more pain and struggle. And that's what hurts about it to me, is you got to have some empathy for these people, that they are making a change that's really important in their lives, and it's not so that they can so that they can beat some girls at a sport. It's because this is part of their life, and we've got to respect that. Okay, I'm rambling on. I said I'm going to make a video about this, maybe this weekend. Maybe I'll put the spiders down for a weekend. Well, not for the whole weekend. I got plans for the weekend for spiders, but let's see. If Stephen Jay Gould were alive today, his scientific rigor would be making similar arguments. Yeah, Stephen Jay Gould was ahead of his time. He would be making these same kinds of arguments that against the kind of genetic determinism and now sexual determinism that so many people are making. There's something in our culture that makes it really appealing to fit into a narrow niche, to be able to say, oh, because you have this trait, you have all these other associated traits. And Gould would have freaked out over that. Wish he were still alive. He'd be a good one to be in this argument. Let's see. I remember reading in a 100 level bio course, there's a type of creature, I think it was a type of algae, which has 12 to 13 genders. Oh yeah, there's lots of organisms that have more than two sexes, actually. Uh, yeah, there are yeast mating types where there are, for instance, simply different alleles that give you different compatibilities in mating. So yeah. Let's see. Sam Harris claims that Stephen Jay Gould was a fraud who falsified data. Uh, yeah. Sam Harris is a fraud. I have had enough of that guy, really. Um, yeah, there was an article going around a while back that claimed that and he made a study of these old time phrenologists or craniometrists who studied the who studied the volume of skulls, and there were different methods for doing that. That things like getting sesame seeds and filling up the skull and packing them down really hard, and then pouring them out and weighing them, and you can get an estimate of the volume. Um, and there was an article that, so I think it might have been in one of his books actually. Gould actually said that he repeated some of these things, and it seems like it's one of these things where, depending on how hard you push down the seeds, you can get variation in the volume, which opens the door to bias. If you've got a black person's skull, you don't push quite as hard. And if it's a Caucasian skull, there's no such thing as a Caucasian, except for some people living in the Caucasus Mountains. Anyway, you've got this white person's skull. Well, obviously, it's got to be a bigger volume, so you really jam the stuff in there. And then there was another paper that came out a few years ago in which somebody said, well, no, it wasn't the original study that was cheating. It was Stephen Jay Gould who was fudging the data. I don't care anymore, because what that said is that there's so much opportunity for bias in these kinds of measurements, in these crude kinds of measurements, that that makes the whole data set suspect. What does it mean? Then, of course, you look at individual variation. Within white people, there's a huge variation in skull volume. It doesn't seem to affect intelligence. There's a consistent average difference between men and women. Doesn't seem to make a difference in intelligence. So why do we care about this? What we ought to be doing is documenting all this variation, saying, well, clearly, skull volume is not a determinant of human intelligence, that there are other factors that we haven't yet identified. Okay, let's see. Somebody mentions, yeah, most women do have testosterone. They certainly do. Testosterone has been found to be important in women in their sex drive. And so, yeah, it's a mix of things. And, when you actually look at the biochemistry of these steroidal hormones, uh, there's a whole range of different steroids that are involved in the synthesis, all kinds of precursors and synthesis of testosterone. And each one of these variants has a proportion in the individual. So it's not a simple black or white thing. It's, yeah, women tend to have, very strongly tend to have reduced testosterone compared to men, but they still have some. Men undergo all kinds of changes in testosterone. I've never had my testosterone checked, but I've been curious. It's typical that in older men like me, testosterone levels are dropping. They're getting lower and lower and lower. Oh man, that's scary, isn't it? Oh man, I'm less of a man because my testosterone levels have been dropping, and I haven't even cared enough to go check them out. Yeah, who knows? Anyway, the thing is, it doesn't really make a big difference, right? That you can have these tremendous variations in testosterone levels, and they're poorly correlated with variations in sex drive. When they get below, when they get down to a pathologically low level, you've got to worry about it. Uh, but otherwise, we tolerate and accept a huge range of variation in testosterone. And furthermore, that what that's telling us is that sexual behavior is not entirely determined by testosterone. I eat a lot of soy, so maybe my testosterone levels are even lower than I expect. Yet I still have a perfectly normal sex drive. We will not discuss it here; that's private. But no, I'm not. I'm not neutered at all. So that stuff just doesn't matter as much. Oh, somebody asks what I thought of developmental systems theories advanced by Paul Griffiths, Russell Gray, Susan Oyama, etc. Oh, I love Susan Oyama. I've got all her books over here because I really got into DST for a long, long time. Uh, Here's the catch, though. DST is a philosophical position. Just the fact that Susan Oyama and Griffiths are both philosophers, not entirely working biologists, means it's a little bit disconnected from what we think of when we think of doing as biologists. And that was always the frustration: is here's this great conceptual idea. Here's a wonderful way to look at how development and evolution interact. Yet when I go into the lab, I I have a hard time getting a grip on how I'm going to use that stuff. So, what you find in the field right now is mainly Evo Devo, which is uh, which is not as philosophically deep as DST, but it is much more grounded in the practicality of what we can do in the lab. So with Evo Devo, you spend a lot of time, looking at proteins and gene sequences and comparing them, and all this kind of stuff to get a feel for degrees of propinquity and build trees and all that kind of cool stuff that evolutionary biologists do. And DST, no, I just found it. It's one of those things. This is one of the things I wanted to mention in this video. I was planning this weekend, but it will probably get cut because there's limits to how much I can pack in there. Is that um, there are philosophical presuppositions and philosophical concepts that inform how we interpret the data. And I consider a developmental biologist to have an obligation to at least read some of this stuff, because it's part of the background environment in their brain for how they interpret information coming into it. Uh, so that's mainly what I see as being important, is that we build a worldview based on a breadth of data that comes from lots of different fields, and things like philosophy are an important input into how our brains operate. And too often what you find with working biologists is, no, they disregard philosophy and they think it's a waste of time. But it's really not. It's how you shape how you understand the world. So that's my perspective on DST. Even though I don't have a good, strong way to use it in the lab, it's something that always makes me think. And for instance, my position on trans issues is informed by my understanding of developmental systems theory. So, yeah. Let's see. Scientists are philosophers with better data. Sometimes they're bad philosophers with better data, though. That's always my concern. You know, there are people like Hawking and Krauss, who were big name physicists who then came out and declared that philosophy was dead and useless. That was embarrassing. Uh, yeah, like people are saying, science was originally called natural philosophy. Science is philosophy. And when you say philosophy is useless, then you're saying that science is useless. So, okay. Question: does what have anything to do with a bias? I don't know. What are you talking about? Oh, yes. Krauss was doing philosophy while he was shitting on it. Yeah, that's too true. Yeah. You know, here's another awkward thing, though, is I am not a trained philosopher. And so when I say philosophy is really important, we ought to know more about it, I'm speaking as a fairly naive individual within the field of philosophy. Uh, all I can do is say, yeah, I'm going to give props and respect to the people who bring philosophical perspectives to my science. I'm not going to disregard them. I don't think I could ever, give a clear and competent um, new philosophical perspective. So I will happily incorporate all these ideas from philosophers and scientists and see what will happen. ADX mentions, I thought natural ebbs and flow of hormonal levels are generally accepted within humans. Yes, they are. That's the other thing. You may think you're a manly man, but your testosterone levels change all the time. Uh, Same with women. They go through a monthly cycle even where hormonal levels change. So, yeah. Also true in animals. Uh-huh. Okay, how is this for a compromise? Philosophers make hypotheses that scientists can go out to experiment. Yeah, I agree with that. Um, Here's my one problem with that is that, when you're deeper into the science, what you discover is that most of the stuff we're doing is fairly fine grained. We're looking at small distinctions between different testable hypotheses. So that just means, in a lot of ways, when I read philosophy, there's this grand scope, this that's putting everything into this giant perspective. And realistically, when I'm sitting in the lab, what I'm doing is I'm looking at little things like, oh, are Texas spiders more sensitive to the cold than Minnesota spiders? Yeah, so it's part of the bigger picture of the story, but scientists are typically working at a fairly fine scale on little problems. So yeah, I just hesitate to say I'm establishing new philosophies here. Yeah. So let's see. Oh, that's a good point. Science is an established method, whereas philosophy questions method in epistemology. Yeah. Um, this is another thing that in science, uh, where you succeed in science is when you implement a method really well. So somebody works out a technique and then you grind it out and you get lots of data using this technique and you publish it and you get famous and you get grants. That's what we do there. Um, at some point, somebody steps in and says, "But wait a minute, let's consider the actual implications of what you're doing. Is this really valid?" A good example would be evolutionary psychology. I am forever shocked at all the people who are churning out evolutionary psychology papers that are total crap. Um, they really need to step back and look at the field as a whole and compare it to what evolutionary biology says, because there's a gigantic disconnect. That would be a good point where some philosophy of science would help. Let's see. Oh, that's a good question. Are you going to have to inbreed a lot to get any kind of basis with spiders? Couldn't this be a problem? Then you have to bring in regular mates from the outside. Yes. Then I've already been concerned about that. So last year I collected a bunch of spiders and I was breeding them over the winter. And one of the mistakes I think I made is that I was really gung ho, and from my familiarity with zebrafish, I said, what'd be really useful is to have a nicely inbred strain, which was a little more predictable in its outputs. And I started breeding mothers with sons and so forth, and just really intensely inbreeding. And what I discovered is, by about the third generation, they weren't doing so well. So, oh, and thank you, Joshua Schroed. I don't think I've ever gotten one of those before, but I appreciate it. Thanks. Anyway, so I was inbreeding them, and then in the middle of the winter, I was discovering that I was having all kinds of problems getting egg cases and progeny, and it was just, and I think it was because I inbred them. They were not doing very well, and they weren't ready for this. So yeah, this time around, I'm going to start with a larger colony, and I'm going to track progeny, and we will. We don't know the ancestry of these spiders, so they could be related. Some of them certainly are, but I'll at least try to get some diversity and do some outbreeding first, because I want a stable colony instead of one that peters out after 6 months. Okay, how do I explain Noah? Noah's not real. That's how I explain Noah. Yeah, ancient cultures were very bad at genetics. Read the Bible, and they have no idea. But it's not just the Bible. Anything before the 19th century, they were really bad at genetics. Darwin was terrible at genetics. He was struggling to understand how inheritance works. That's why Darwin ultimately settled on Lamarckism as his model for genetics. Yeah, what we call Lamarckism is actually classic Darwinian genetics with gemmules and all this other stuff. It was just, ugh. Let's see, didn't Mendel attempt to contact Darwin directly? Well, sort of. Uh, yeah, the history is just kind of complicated there, but yeah, Darwin published The Origin of Species about the same time that Mendel, within a few years of Mendel publishing his paper. But it was shortly after that Mendel sort of retired from genetics and focused on being the abbot of a monastery. So he kind of stopped working on that stuff shortly after. So I don't think he applied any pressure to it. I heard, though, that on Darwin's bookshelf was a volume of the book that included Mendel's paper, but it was uncut, so he'd never read it. So, but lots of people read that before 1900, and nobody made the connection. If you've ever read Mendel's original paper, it's a snooze. It really is. It's all statistics and numbers. It's just lots and lots of crosses with tables and tables and tables of the results. He's got a few very clear-cut conclusions, about the ratios and all that sort of thing. But I don't think Darwin was mentally prepared to listen to it. Oh, uncut. I would not have thought to hear that about a book. Yeah, in the old bookbinding method, when you bought a book, what you got was a book where adjacent pages were folded over, but they weren't cut up there at where you turn the page. And so when you read a new book, what you would do is you would sit there with a letter opener and you'd rip and split all the pages as you go, which was kind of neat, because then historians can take a look at some old guy's bookshelf and see which papers he read and which he didn't. Yeah. Nowadays, of course, they bind them the same way. There's the same paper folding technique. Then they go through these massive machines that have razors that come down and slice everything for you, which is much more convenient. It's like sliced bread nowadays. Okay, let's see. Professor Frank says he's a geographer. Geography should be both microscopic and macroscopic. Yes, geography is really important. Yeah, we were just looking at our data, our spider data for June, and thinking, hey, we got to start mapping this stuff, because we saw these weird things where, as I mentioned earlier, generally what we find is Parasteatota and Fulcus everywhere. But what we also discovered is that there are little islands. There are places where we find, for instance, that Steatota triangulosa I mentioned. There was one garage that was just full of triangulosa. And so what I want to do is I also want to map this stuff out and see if there are distinct subpopulations, like ethnic neighborhoods in Morris, where you find certain species or subspecies localized, and ask, do those persist over the years? So yeah, that's a kind of, it's spider geography. We'll look at spider geography. That would be cool. Yeah, nobody has letter openers now. Well, I think I've got one. They're useful when students get rambunctious. You just whip it out. No, not really. Reading Darwin was cool because he debunked all the intelligent design arguments in the third edition, including irreducible complexity. Yeah. So I read Darwin's Origin of Species concurrently with what's his name? Who was the big natural theology guy who Darwin actually liked a lot when he first went on the Beagle? He wrote a book called Natural Theology, and I read that. And it's really amazing. You read those two together, and what you see is that Darwin is specifically addressing the points that this guy brought up at each point in roughly the same order through his book. The Origin of Species is basically like a great big response video to a theologian with data. So it's good stuff. Yeah, Paley. Yeah, I don't know. My brain is, I'm getting tired here. I spent a long day with the spiders, and now I'm starting to fade out. But yeah, he's comparing Paley. I also like the fact that Percy Shelley debunked intelligent design before Darwin even. Yeah, you ought to read more Percy Shelley. He's amazing. Oh, microclimates influencing populations. Yes, I'm sure of that. So one of I keep spilling the beans about what we're going to be doing, but you guys won't scoop me. None of you are going to rush to Morris. And anyway, one of the things we were noticing is that when we go in different garages, because of different degrees of insulation, how much they're open, and all this sort of things, there's clear variations in temperature. So there were some garages we walked in, we said, hey, this is great. This is cool and comfortable. What's going on here? It's not air conditioned. It's just, the orientation and whether there's trees providing shade and all this kind of stuff. And then you go into other garages and you say, oh, this is a nightmare. Get me out of here fast because I'm breaking out in a sweat. So that's another thing we're going to record, is these temperature variations in these different places. And this would be another correlation to look for is, hey, if it's a warm place, does it have a different population of spiders than if it's a cool place? We'll find out. It's one of the nice things about this study is we just keep thinking of things that we can easily look at once we get this pile of data after a long summer of hard work. So we'll be working on it. See, I always thought evolutionary biology was the stuff about how we became social. So the evolution of eusociality is one small aspect of evolutionary biology. There's a lot more. So I think you're just talking about a subset of evolutionary biology. Yes, and temps have been increasing overall. I don't know if you were here earlier, but I mentioned that's one of the things we're going to be looking at too, is this is a baseline year. We just figure out what's here now. And then we'll come back to these same places in subsequent years and ask if they've changed over time. See, Cratius make Lamarck and Lysenko look intelligent. Lamarck was sincere. Lamarck, yeah, he was doing real work and he was really thinking. So it's not fair to say that about Lamarck. Lamarck was cutting edge for his age. Just that we went well beyond that. Lysenko, on the other hand, was a political hack, and he distorted the science to suit his desired ends. I would not compare Lamarck and Lysenko ever. Oh man, all these hard questions are now starting to come in. Is it possible to hardwire greater empathy into our behavior as a species? How are you going to do that? Are we going to CRISPR-Cas everybody and insert? No, that won't work. No, that's the only way to do this. Culture is much more important than genetics on that. And I think what we just need to do is teach people better, and that's how we'll get greater empathy into the population. Is catch little kids early and teach them how important it is to care about their friends and neighbors. Okay, you only have spider webs in our 7th floor flat over the stove, and they're all covered in oil vapor, and I cleaned them up a bit. But really have no spider. Oh, that's weird. Covered in oil vapor. Huh. Are you sure that's the only place you have them? So one of the tricks we use when we go out in the field looking for spiders is we bring a little spray bottle. Oh, this is a small one. But we got bigger ones. A little spray bottle, and you just spritz that and mist region where you expect there might be a spider, and it makes all those cobwebs visible. They get little beads of water on them. So maybe it's just you're seeing them because they're getting oil vapor, but elsewhere they're there, but you're not seeing them because you're not coating them with anything. Also, we get what amazes me is the other day we made the first recorded observation of Fulcus manuelli in Stevens County ever. And we discovered it in the science building. And the science building, it looks like a hostile environment because our custodians are really thorough, and they come through every day and they clean everything, they dust everything. Cobwebs do not last in that building. Yet what we found was a huge specimen, a huge female, just wandering around down on the ground floor, looking good. So we know that they are just clever about hiding in various places. I kind of suspect, we have those ceiling spaces where all the ductwork and the wiring and everything is. I bet you those are just spider paradises up there. And it's just that every once in a while one descends down and then we see it. 8DX just noticed another spider next to the balcony door. Yeah, once you start looking for them, it's amazing. Yeah, you start seeing them everywhere. Years ago when I was fossil hunting, I noticed this as well, that you go out into this dusty brownish gray field and you look around and you're saying, there's nothing here. And you got an expert who comes along and shows you, oh, there's one there and there's one there. And, here's a little bone fragment. And you start suddenly picking up on that stuff and you wander through there. And all of a sudden it turns into this giant cemetery. There's bones everywhere. And it's the same with spiders. Yes, I have seen Australian peacock spiders dance. They're lovely. Even the little ones we got here. Oh, you're not going to be able to see this. Let me see. Yeah. See there? That's a zebra spider right there, scurrying around. Those dance as well. Maybe not as colorful as those peacock spiders, but they've got pretty much the same dance where they come up with the female and they raise their forelegs and they kind of scoot around and they show off and they wiggle their butts and all this kind of cool stuff. Uh, there's lots of fun there. So yeah, they do that. Yes, I have a few spiders in my office right now because my wife has turned out to be really good at spider hunting. And she's out there gardening every day. And she finds a spider and she scoops it up and brings it into me. So let's see. Personal question. How did you come to be so intolerant of fools? Why should I be tolerant of fools? That's irrational. Anyway, I come from a painfully agreeable family and just envy your superpower to shame morons. Oh, uh, now you're getting into a gray area, a problematic area, because some people will tell you that I'm terrible at how mean I am to foolish people. Uh, where does it come from? Well, that's history. I was fortunate. I grew up in a lovely family. We didn't have weird, horrible religious views or weird, horrible political views. Even even now, when I get together with my family back in Seattle, we're all on the same wavelength. We're all basically radical leftists. So I grew up in this very supportive environment where we got these ideas. My father was a fanatical labor unionist. And why was he? Because he cared about people, that unions were the way to beat corporate power and all that kind of stuff. So there's a strong installation of empathy to go with that. And what I'm really intolerant of is those people who lack that empathy. And we've seen them come to the forefront nowadays. But let's see where I came from is I went off to college. I was the first person in my family to go to college and started learning all this wonderful, exciting stuff. I was so happy to be learning biology. And then I encountered these crazy people who believed in creationism. And it's like encountering hostile aliens. You want to set the phaser to stun and just get rid of them. Uh, but you can't do that in polite company. So what you have to do instead is argue with them. So yeah, that's where it came from, is being steeped in evolutionary biology. And then this is one of the flashpoints in the culture wars, is evolution, and meeting people who were clearly ignorant and yet so confident that they were right. Just read Ken Ham now and then, and it's the same thing. So that's why it is. Yes, I can. Well, I'm actually from a suburb of Seattle. I grew up in Kent. If you're in Queen Anne, you probably know Kent down south. Yeah, those of you who don't know the Seattle area, there's Seattle, and then there's this chain of little satellite suburbs dangling down below it. There's Renton, and then Kent, and then Auburn, and they're all Boeing towns now, although Boeing has been losing its influence, which I think is a good thing. So there are all these Boeing towns scattered around there. So the Puget Sound region was just a string of company towns, and yeah. It's one of my father's great dilemmas. Was he wanted to be working at Boeing because that was the only place he could go to get a decent wage and to get things like decent dental and medical care. And yeah, he didn't really like Boeing. I don't know anybody in that area who likes Boeing in the sense of regarding them as a kindly uncle that can give them all these opportunities. It's more like here's this horrible skinflint that you got to struggle to get money from. Yes, Kent. Oh, I was back in Kent the year before last. It's greatly changed from my childhood. When I was a kid, it was a lot like Morris, where I am now, small town. And now it's much more suburban. There are some advantages there. I remember when Kent had one movie theater and then it closed down, and every child in town wept because now there were no more Saturday matinees for us. And now they got an amazing mega theater there in Kent, which, little pluses there. Oh, it does. A hot housing market, huh? Oh, I'm not in the market, fortunately. I'm not planning to move for a while yet. Uh, okay, Eric. Oh, Eric Hovind is more insufferable than Kent Hovind. Oh, that's a toss-up. Have you seen Kent Hovind's YouTube videos lately? He's still making them, and they're awful. They're just Kent Hovind sitting there in his own usual style, and just, oh man, awful. But yes, Eric has been more infected with presuppositionalism, so uh, they're all terrible people. Yeah, Kent is Kent. Is Kent way worse than Eric Hovind? I don't know. The only way to settle this is with a cage match. That's what we need. Throw the two of them together in a cage with. We can have chainsaws and knives and swords hanging from the sides, and just let them get on each other's case and see what happened. Yeah, Kent shaped Eric. So there's the other dilemma, is that when you're talking with a creationist, you are talking with somebody who has been wrecked by religion. They may be saying stupid things, but in a lot of ways, it's not entirely their fault. There was somebody who poisoned them. I'm surprised you didn't run into more creationists before college. Oh, well, this was in the 1960s and 70s, okay? And creationism, the creationism of the form we have now really got started in 1961 with the Genesis flood, um, by Whitcomb and Morris. And that gradually expanded to take over. When I was growing up as a kid, this was non-existent. Nobody talked about it. When I was going to high school in the 70s, it was more like nobody took that stuff seriously, but they still avoided teaching evolution in the schools because nobody wants the crazy guy from down the street to come in and yell at you. So there was this growing pressure that I could detect even then. And what it meant is they didn't teach creationism, they just didn't teach anything in that subject at all. So yeah, I didn't learn any evolutionary biology in high school at all. Not one bit. Didn't even mention the E word in my science classes. Of course, I was reading it on my own, which helped, including reading Stephen Jay Gould, who was very influential. And then I got into college, and of course, day one of biology class, it was, yeah, here's evolution. You got to know this to understand what's going on. And it's the same thing now. You come to college here, day one, your first freshman biology course. The first one we offer is called Fundamentals of Genetics, Evolution, and Development. You're not going to escape it here. Yeah, a lot of people thought creation science was science because They worked so hard to put up the illusion that what they were doing was scientific, even when it wasn't. So, oh yeah, okay. Joshua Strode, his dad raised him on an odd mix of creationism and physics. Yeah. So, did you see that post I put up the other day about Danny Faulkner? Uh, so, Danny Faulkner is he works with Answers in Genesis. He's a creationist, a young Earth creationist. His background is in astronomy, and it was the weirdest article because what he's doing is he's talking about how crazy it is that people believe in a flat Earth, and here's all this scientific evidence to demonstrate that the Earth is a sphere, etc. And he's going through these flat Earth arguments, and he's basically saying, why are you discarding all the knowledge that we've acquired over thousands of years and all this information that's available now? And, it's a kind of hubris to discard the science of, you know, physics and geology. And it's just so discordant because this is what he does to biology, is he's throwing out biology for his old myth. Um, yeah, Abby Hafer is a not so intelligent designer. Yeah, that book it's a good book, and she's got a new book out. She's working on a new book, and her publisher is going to send me one any day now. I'm rather anxiously awaiting it so that I can review it. Yeah, she's smart. She's funny. Um, I'm really looking forward to that, and I'm glad she didn't stop at just one book. There's more coming. Yeah, Abby ought to be invited to more of these atheist skeptic conferences. This is one of the most depressing things about the state of the atheist community is they discard so much valuable stuff because if it's not an old white guy, it's not worth hearing. Yeah, so and I say that as an old white guy. I got the credentials. See, doesn't ID require God to be the most obsessive, compulsive control freak imaginable? Yeah. Yeah, did you read, what's his name? Oh, man, I must be getting tired if I'm blanking on Behe's book. His book is just, in his second to the last book, he's basically saying that any trait that requires two concurrent evolutionary changes is mathematically impossible. It's statistically unlikely. And his example case is resistance to malaria. That are malarial resistance to things like quinine and that what he's saying is it would have required the malarial parasite to acquire these two independent mutations, and the odds of that are astronomical. Therefore, they must have been designed. And when you read through it and you think about the implications, yeah, he's doing the math wrong, obviously. But the implications of that interpretation is that God is noodling everything, that God is specifically going in and fine-tuning the malarial parasite to do a better job of killing humans. And that it's true of all the diseases. They've all got multiple mutations. We can track the AIDS virus and how it changes within an individual over time. It's constantly acquiring mutations. And those mutations are one of the things that makes it hard to defeat it. But you can actually trace those. And what Behe's interpretation would be that God is looking at that AIDS victim and he's reaching down and he's tweaking that virus to avoid any kind of treatment, to be more effective, to do more harm to that individual. It means that God is not just obsessive, but he's downright evil. And he's really interested in harming people. Yes, astronomical things happen all the time. And what Behe doesn't seem to consider in his models is something as basic as recombination. That one of the ways you get, you can have mutations arising independently in different organisms, and then through recombination, you get them into one individual, and then you've got your two chance mutations occurring together. And yeah. Yeah, science education journalism channels still exist, but most have dissociated from that community. Yeah, you know, years ago, if you had a science channel on YouTube, some of them were by Christians, of course, but a lot of them were by secular folk, and they were not shy about saying so. They were not at all embarrassed about saying, "Yeah, I'm an atheist." They were not judgmental and say, "You can't listen to me unless you're an atheist too." But they were pretty open about this sort of thing. And nowadays, if you've got a science channel, there's a few of the more aggressive atheist ones that will speak out about it. But now, if you want to talk about science, your best bet is to avoid that whole slimy pit of atheist noise. And that's just sad. Yeah. I only talk about it because I'm a relic, but oh well. Oh, and look at this. We've been going for 2 hours. This is too long. I don't listen to videos that are 2 hours long. So I think we should stop here. But yeah, thanks everyone for joining in, and thanks for the good questions. I'm going to try and get a scripted video up this weekend. I have plans. My plans often fall through, but I'll try to get that going. And I've got to figure out how these hangouts are going to work in the future because they're changing everything. And I'll try to do one. I'll probably be doing another practice one in a few weeks, just trying to puzzle out how the heck these things work in the future. Okay, sorry, Silicon Baby. Yeah, you're late. Too bad, but it's all being recorded. You can go back and watch it. Okay, well, I think we should stop there, and I should go rescue my wife who's out there watching the Democratic debates. Notice how clever I was to schedule this for the time of the Democratic debates so that we'd have an excuse not to listen to them. Okay, so good night. I'll try not to be so neglectful of my YouTube channel in the future. Talk to you later.